Hello, everyone. My name is Yuang. I'm from Ecology and Evolutionary Biology. I think many of times I came here to grab the pizza and enjoying the presentation for this kind of tools and technology seminar. So an idea came up to my mind. My thoughts came here to give a presentation today. So I'm here today. Uh, I'm glad to be here. I'm studying the fitness landscape. So, what is fitness? In the evolutionary genetic, the fitness is the ability of an organism to survive and reproductive in a specific environment. So different environments could have different kind of the fitness, and it will create a fitness landscape for the whole organism. So let's take the saccharomyces for examples. So it will be the growth rate for saccharomyces is in the 1 PD nutrition plates or liquids. So we could also design the specific plasmid or enzyme. It could be the catalytic or ability to survive in a certain toxic environments. So what is a fitness landscape look like? And I think the landscape is for each genotype assigned with fitness. And when we visualize the landscape, let's go to the last slide. Each genotype, they are collected. So there are, let's take the 3 sides, for example. Each side have 4 states, ATCG. So the landscape side is 4 to the 3. And for each node, they are collected because there are only one size difference, So here is the collected electron. And for the different color, it indicates the fitness levels. You can see the yellow is high fitness, and the dark black is a low fitness. And when we visualize the 3D structures, you can see the x-axis is a space, a sequence space is a genotype, and I think the height of the mountain landscape is fitness. So understand the fitness landscape. We could understand to investigate the evolutionary trajectory for species, for organism. So it might be smoothly only with one peak. It might be multiple peak and highly rock. So we could using the fitness landscape to predict evolutionary trajectory and understanding the mechanism of adaptation. So let's take the Ecology for A gene for examples. And here is Ecology dihydrofolase reductase. This gene has one domain. It combined with 3 important amino acid sites, and for these sites, we could because there are 3 amino acid sites, and it's corresponding to 9 nucleotides. So we could create mutant genetics by the CRISPR-Cas9 gene editings. And after you can combine all kinds of combinations. So each site have 4 states, ATCG. So there are 9 sites. So the landscape size is 4 to the 9. And after you create this kind of library, and we could using the mass selection experiments using antibiotic environment to selections, which genotype or which variant is favored or is more help to living in the antibiotic environment. So for the created library, we could do the deep sequences here before, and each of variants it have the variance frequencies by the Illumina sequences, and after the selections and the antibiotic environments. So some of variants they are not survive. They are not beneficial. They don't have the ability to catalyze the toxic environment, so the rate may be decline. And some of the environments they might be some of the variants they might be beneficial. They could survive. So the variance may be a lot. So we could using these two numbers variance, the variance frequency to calculate the fitness. And because of the variance, they are connected with several steps differences from each other. So we could using this kind of library to generate the fitness landscape. And when we realize it, and the 3D structure like the landscape like that. And for the landscape, we have 4 to 9 variants. So what is the adaptive work? For given the left for given landscape, and we could start with random variants, just like the blue color, blue dots, with random choosing a variance or genotype, and every time we selection for the high fitness. So doing this kind of adaptive work, and we want to know after the selections, because evolution only favors high fitness. After the selections, the variance will end up with a peak. So how frequently each of the peak. So we will calculate the peak's fitness frequencies. You can find if each of the peak is defined by the specific genotype, but their fitness is higher than their labors. So if we calculate the frequency of peak fitness, suppose they are equal, so the frequency is the same, and we based on the peak fitness, we'll calculate the cumulative frequencies. So it should be like the linear way. But actually, from the landscape, the high peak is supposed to have more attractiveness. So it's not supposed to like in the equal frequency. So why should we learn the fitness landscape and the adaptive work? And from these two concepts, we could understand the evolutionary dynamic and the adaptive evolution and how the genetic variants influence the organism fitness. And by the adaptive work, our question is that the population always reach to the high fitness, and or when there are lots of low peaks, just like this two, there are be trapped in a low peak a lot. So studying the adaptive work, the beneficial mutation will lead to the fitness peak. So we could understand whether evolution is deterministic or just somatic, just by the random. And for the Ecology for A genes, the created fitness landscape library, and we do the adaptive work, and the axis is peak fitness. We simulate thousands of times. Each of times we randomly assign a random sequences at the starting point, and every time for each of adaptive work, we selected for the high fitness compared to the original one. So finally, it will end up with the peak. So the peak fitness is is higher than their neighbors. And here is the cumulative frequency, just like this shape. So, when we set a cutoff, because based on the peak fitness, we could define the high peak and the low peak. We select we set cutoff top 40% of the peak fitness at high peak. So reaching to the high fitness peak, it's accounted for about 76%. So that's amazing. I think only top 14% of the peaks have larger attractions. There are attracts about 70% of the reference. So, but when we define the high peak, there is some arbitrary, because from these studies, they have the position 27. There are lots of in the green and the yellows, and the panel is the peaks ranked by the fitness. So from the right, the more have the high fitness, and the most peak is the blue, the green and the yellow ones. So they define the and at this position, the blue, the green and yellow one is aspartic and glucamic. So, so when we have this kind of adaptive work, we found the peak, the frequency of which the peak is so different. So the variation is not relevant, because a high peak with high frequency to be end up with. So, and for this study, the top 40% of peaks is attraction about 70%, 76% of the reference when we assign a random reference. And when we calculate the top 40%, we could take the top 1 or top 2, top 10, for example. Here is a cumulative frequencies. And when we calculate the top two peak, and here is the cumulative frequency, that means this top two, they added up is their top two peaks attractions. So yeah,
just I'm perpetually confused.
Mm-hmm.
You have two illustrations on that surface chart where your adaptive walk
Mm-hmm.
seems to go from what looks like a peak across a valley
Mm-hmm.
to another peak. Well, if crossing that valley means that your frequency is going to be very low, isn't there a chance that it's going to go to 0 and then the adaptive walk will stop?
In the adaptive walk, we only select for the higher fitness. This is the model, just say the trajectory, what that trajectory look like.
Okay.
Yes, we only select for the higher fitness compared to the original one. So when it go down, when the fitness go down, it will be stopped.
Oh, okay.
Yeah. So our question is whether this trend still hold in different kind of in wide range of landscape. What trend? The top 40% of peaks have a large have 70%, over 70% of attractions of the whole reference. So we collected the different kinds of landscape. Let's look at this studies is investigate the regulatory abilities. It calls shiny Dactylos sequences. It could accurate the translation initiations. So from this study, in the they could design the SD sequences and to see the downstream the GFP, the expression level. The GFP is a marker gene to detect the regulatory abilities. So the landscape also have four states and have 9 sites, and each site have 4 states. And here is the adaptive work look like. and top 40% is the right is top 40% of the peaks it end up with. So for the probability is also over 70%. The trend seems to be hold. Let's go to another study. It's a tRNA. The tRNA in the east, and it has I think it has 10 variable sites. The first 6 variable is each side have 2 states is binary, and the rest of the 3 sides is have 4 mutation mutation probabilities. And for this landscape is combine here is the landscape size. So we also simulate adaptive work, and here is the frequency of adaptive work look like. So when we set the cutoff top 40% of the landscape, and the frequency of reaching high peak only 70%. So the trend seems to be not cold. And we selected a lot of representative landscape, for example. And here is the SARS-CoV variants. Here is a key domain, the RABD, and we want to understand the DNA sequences with the potence, the binding affinities. So here is the landscape, and here is the adaptive work look like. So reaching to the top 40%, it has the frequency of reaching to is about 80%, over 80%. So it seems to be high. And we also selected a fluorescence protons. In protons, we put mutants, 30 mutations. Because there's 30 mutation, 30 sites are important for the fluorescence protons. So for landscape is due to the 13 for the variant sites, and for the adaptive work it's only 30%. And the last landscape we're choosing is from proton G domain. It's only proton landscape because it only have 4 sites, but each side have 20 states because the proton it has 20 amino acids. And here is the landscape look like, and you could find and a lot of adaptive work. They are finally reached to 40% of top peaks, and there are 90%, over 96%. So let's global say the whole picture of the landscape we're choosing. The first is in the Ecology, the DHFR genes, and the dash line is the cutoff for the defined high peak. And we could find reaching to high peak, the probability seems to be different across the 6 empirical landscape. So that means reaching to high peak is not always with high probabilities, because there are some only 30%, and there are also 30%, but there are some protons landscape 90%. So the first conclusion we think is the probability to reaching high peak variance widening across the empirical landscape. But you could find the landscape there are the defined fitness that's so different, and the landscape size that's so different, and the number of the variants, because some could be the protons, it have 20 states, some could be binary mutants, a lot binary, and some with 80 the DNA sequences. So it seems to be there are lots directly comparable. So we're using the theoretical landscape. This one is called Rock Mountain Fuji. The name because it came from the Japan mountain, Fuji mountain. And For this mountain, it only have one peak. Yes, it only have one peak for this landscape. And when we simulation when we want to simulation the Rock Mountain Fuji landscape, we select the highest fitness is 1, and of course all the fitness from 0 to 1. And we introduce formulas, and for given highest fitness, and it is a global peak, and the rest of the fitness is based on the Hamming distance. That means the number of sides are different, and their fitness might be different. And we introduce the random fitness effects by delta, and we delta is this, and this is then deviation. So arbitrarily introduce the variant to simulate, create a lot of high rock or low rock landscape. And B is when delta increase, and C when delta is close to 10. And because people found this landscape is highly rocked and lots of local peak except for the global peak. So it has a high probability when we adaptive work, they're easy to be attracted in the local peak instead of the global one. So we do a lot of simulation based on the different delta. When delta is low, there are only 500 peaks, and when delta is close to 10, and the number of peaks is nearly over 9,000, so it's 20 times. So it's a good simulation. So when we say the reaching to the top 40%, the probabilities, they are from 96% and decrease, and then it decreased to 8, 18, 17, 16, and here it decreased 26%. So the delta matter because delta could create a lot of number of local peaks, and the shape of the adaptive work changes because when delta is low, and there are a lot of small peaks, so there are traction in there's attraction in a small peak, but the frequency is not so high. And and you could say the global peak also have a sharp increase because they still have a lot of attraction for the high fitness peak. And when you directly say the delta is 10, just like this, so we can say a lot of local peaks, they are increasing, the frequency increasing sharply. That means a lot of adaptive work, the variance are tracked in the local peak instead of the global optimal, because they are tracked. Let's go ahead. So the figure A is the example to say when delta is 0.5, and when we say the delta is 0, there are only one peak, and there are only one peak. The number peak is 1. Yeah, and we do this correlation for the delta. When delta increase from the 0.5 to 10, the probability of reaching high peak decrease. It decreases a lot until 20%. Yes, until 20%. And here the R is minus 0.7. And you will find the delta, when the delta increase, the number of peaks also increase. So number of peaks matters. So we calculate frequency of genotypes that are peaks. We're using the number of peaks divide the whole variance of the landscape. So the probability decrease a lot by by the number of peaks increase. And here is a picture of the landscape, mountain landscape. You can see there are lots of peaks, and they are so rugged, so reaching to the high peak seem to be attractive. And this picture is actually from my friends when they go to the Arizona, they take flights, and they said this landscape is so rugged, it's very similar to your studies. Yes. And we give a summary. The landscape ruggedness strongly influence the probability of reaching high peaks, with smoother landscape allows easier access to the high peak, right? So let's go back to the six empirical landscape. There are landscape, the landscape size, they're different, and the number of variants are different. And we want using the Rock Mountain Fuji model to simulate, to understand what the factor for the empirical landscape to contribute to reaching to high peak with high probability or reaching high peak with no probability. So we simulated as the same landscape size and same number of variants and same the states, and we could find the x-axis is a fraction of number of peaks because it is a key factor to determine the landscape ruggedness, and the y-axis is the probability of reaching high peaks, and the different dots indicates a different cutoff because we could arbitrarily choosing top 5% or 40% or top 20% as defined high peak definitions. And here is when delta increase from 0.5 to the maximum, and the number of the probability decrease a lot. And we could find the probability of in this Ecology landscape, they are 70%. One of the reasons we think is because of the landscape is smooth, so they could increase to high peak with much easier. And some of the landscapes they are 70% because the landscape change the ruggedness of landscape change just like this, And when we want to know the protein landscape, what does the why protein landscape has high over 90% of probabilities? We found the landscape is so smooth, so a lot of the variants are attractive with the high peaks, so the probability is very high. So the landscape ruggedness could be directly define the directly factor to determine reaching to the high peak with high fidelity or low fidelity. Because of highly rugged landscape, it is easy to be attracted to the local peak, but actually the empirical landscape does not consistently with the higher expectations, because when we say the red color of 5%, it's not simulated very well found in these situations. Yes, because the shape of landscape, they are also different. So in using the empirical, using the theoretical Rock Mountain Fuji models, the landscape does not consistently inhabit very well. And as for the adaptive work, another factor is the basin size for each landscape also matters. What is the basin size? Let's take the peak A for examples. The peak A is high and they have lots of basin. Basin is the number of the specific genotype that could by the adaptive work they could increase to the peaks. So the peaks A have lots of basins. basin is the genotype that for the adaptive work to reach to it. So each of peak, they have different basin genotype, and we could calculate the basin size. Basin size is define the number of genotypes that could reach to a specific peaks, and divided by the whole genotype, the whole size of the landscape. So and here is vanning. Vanning is so if you start with vanning, is go to the A, go to the C, and go to the D. So there are the vanning is sometimes is regarded as the shared basin for the peaks for the A and E and D, just like that. So the idea came out. So large or high fitness peaks is supposed to have the high basin size, so it has a larger attractions. And we calculate the 6 empirical landscape with the basin size, and the x-axis is the peak's fitness. So each peak, it have a lot of variance that could reach to the peak by the adaptive work. The basin size is the number of genotypes that could reach to the peaks and divided by the whole landscape. And you could find the correlation is positive and is high. And here I put the probability of reaching high peak is 70%. 70%. The correlation is high, 0.8, and 0.7, 0.9. But the 0.9, the correlation does not guarantee for the high frequency duration high peak. You can find there only 70% of the variance that could reach to the high peaks. So for this idea, we introduced the rough mountain Fuji model for the comparisons. And here is the results. When delta is 0.5, there are only 500 peaks. So the correlation is fair like this. And the peaks fitness and the basin size like that. And when and the global optimal, it can find the basin size close to 1. That means except for the peaks variance, a lot of the variance, genotype variance, they could reach into the global optimal by adaptive work. They are close to 1. And when you increase in the delta, the number of peaks also increase. So the landscape become highly rugged, and the correlation decline. For the beginning, not decay so much, and then decline. And when you finally set the delta is 10, highly rugged, and you could find the basin size for the global optimal, it seems to be a lot of their trapped into the local peak instead of the finally reach to the global optimals. So we summarize the results. The next is set an example for when delta is 0.5, and the delta is the delta, and y is the correlation value between the peak's fitness and basin size. The correlation is between the peak fitness and correlation is this correlation values. We found this elective correlations. So the delta increase, the duration to high peak decline, and the correlation between the peak fitness and basin size also decline. It decline a lot when they approach to the highly rugged landscape. So here is our this part summaries. So higher peak generally have larger basins, but actually it does not guarantee or ensure reaching to high peak with high fitness. Yes, you can see from the empirical landscape, the correlation is high, but reaching to high peak the probability is not is actually is unlimited. A lot of adaptive work they are handled by the low fitness peak. So let's go to the summary of results. The firstly, the probability of reaching high peak, they are widely varies across the empirical landscape and the theoretical landscape. We're using the Rock Mountain Fuji models. And actually, in our paper, we are using the NK models and Hausdorff card. Hausdorff card model is the random landscape. And the landscape ruggedness actually they affect a lot about the reaching high peak, the probability of reaching high peak. So with a smooth, with only one or several peaks, the landscape is smooth, so they allow easier access to the high peak. And when we're using the Rock Mountain Fuji model to simulate the empirical landscape, but the empirical landscape does not always consistently inhabits higher than the expected probability of reaching high peak. So large peaks, higher peaks generally have high baseline, but it does not guarantee the reaching to the high peak with high probabilities. And here is our result came out recently published in the molecular ecology, molecular biology and evolutions. And except for that, our result is something limited to the empirical landscape and their limited size, because when you simulation, you could only simulate several genotype sequences. So there's something what they are limited. So we introduce another one is using the machine learning to predict the fitness and using machine learning to construct the whole fitness landscape. And here is a basic result you can find. When we measure the expression levels, we construct a plasmid. The downstream is YLP is a marker gene to detect the promoter sequences, the regulatory abilities, and we could create a 80 base pair. So each side have 4 possible states for each side, and the landscape is so large, and we create a model to simulate and get the results. And using this network, we're comparing the experiments to test whether the model is worked well or not. And and for the results selected for 30 millions of the sequences, and first in one complex medium. The complex medium is the YPD heptones. Another is we could using the same sequences, another defined environment, defined mediums, the synthetic defined medium like URC. So we're using the sequences assigned with expression level. We use this expression level to create the fitness landscape. And here the results look like. The x-axis is a predicted expressions. The y-axis is a measured expression. You can find the correlation is highly correlated, and here the p-values. And the number of dots, number of the testing data is over 1,000 of the sequences. But actually, we are still working on using the machine learning to build the fitness landscape. And but you can find we have also faced some challenges. The predicted landscape, they are larger than the two. Actually, by the measure fitness, measure expression level, they are starting with a 0. So they are not diagonal, although they are highly correlated. So we are also open to any kind of comments or help solutions or cooperations for this landscape construction by the machine learning. Finally, I want to thank my PI George from the Ecology and Evolutionary Biology, and thanks to the NIH, so it could support me to study and do my postdoc training here. Thank you to be there, and I'd love to take any questions. Thank you. Yes.
So for what looked like a lot of your fitness, like proportion of walks to reach a high peak, a lot of the predictions were very gradual rises towards or I guess gradual rises in peakness followed by upshots. I guess I was wondering, why would it be expected that there would be a gradual rise in fitness rather than very sharp stepwise rises all the time, as opposed to as opposed to gradual.
Okay. Actually, by adaptive work, we don't consider the sharply increase or gradually increase, because selection only favor by the higher fitness. So each step we choosing the higher one is okay. So, but I think we could go through our data sets, our simulation results, whether sharply increase is a frequency and is highly frequent. Yeah. Okay.
So
Yes.
one of the online attendees asked, how did you form the trajectories?
The trajectory is, I think the trajectory is based on the adaptive work. Adaptive work is you randomly assign a starting point for a genotype for a given populations. You randomly assign a genotype. It has a fitness. The trajectory is every time you select for higher fitness. So for the same genotype, it might have a lot of trajectory by the adaptive work. Yeah, okay. Answer your questions.
Other questions?
Yes.
I have another question. I guess I was wondering, so like with how you formalize fitness landscapes. I was wondering if this way of exploring fitness can also be combined with the complex dynamics that can occur in like a bacterial community. Mainly because you can imagine that that would be a very rugged landscape just because there are so many possible interactions that can occur.
Yes.
Do you think there's any feasibility with combining this idea of using fitness landscapes and potentially looking at how they can change with respect to more complex dynamics that can occur in nature?
Yeah. Good questions. In this paper, we actually have the epistasis when two sides are interacting with each other's. Suppose with one side, side A, the fitness increase, side B also increase, but when we combine these two sides, the fitness decrease. So it's always is called the reciprocal sign epistasis. It could create a lot of optical to handle this kind of trajectories. Yes. So the when we talk about the rugged landscape, there have a lot of epistasis that affect the evolution trajectory.
I think in the future that could potentially be. Do you think some of those interactions could potentially be like too rugged to explore?
Yes.
Feasibly, or do you think it could be smoothed out by the certain interactions?
Yes, I think so. Because the interaction matters. Interaction is a deep factor to determine number of peaks in the landscape and how rugged it is. Yes.